But give me readers. I don't really know how long now.
So hello guys, we are back with another video. Till now there are only 4 subscribers and I appeal you please subscribe my channel to get latest update. Please follow us on Instagram, TikTok and Facebook. So let's start. Today's topic is oxidative phosphorylation. It is another topic and process of carbohydrate metabolism. It is divided into two parts. First is oxidation, which means donation of electron from a molecule. And second is phosphorylation, which means that addition of phosphate in the adeno diphosphate, which convert itself into adeno triphosphate. As we know that mitochondria is the powerhouse of the cell and most of the processes happens in it. And oxidative phosphorylation is also happens in mitochondria. So let's speed up and have a look at the structure of mitochondria. Mitochondria is basically divided into two membranes. The outer membrane is called outer membrane and the inner is called inner membrane. And the most important oxidative phosphorylation happens in the inner membrane. There are complexes present in inner membrane, some coenzymes, some cytochromes. The complexes are made up of lipids and proteins which have copper and iron. In total, there are 5 complexes, but 4 take part in oxidation. So let's start oxidation first. As you can see in image, the first one is complex 1, then it's complex 2, then it's complex 3, and at the last it is complex 4. Basically, there are 5 complexes present, but 4 take parts in the oxidation. Let's check out what are these complexes and what their process is. So let's start here. We have first with us the complex 1. Complex 1 is basically a flavin mononucleotide, and it is also the center of iron sulfur, which is called iron sulfur center. The complex 2 is also known as succinate dehydrogenase, which also take part in the citric acid cycle, which is also called Krebs cycle. So its activity rise and falls together. The process basically starts in complex 1 and complex 2. Then it follow on into another pump in the inner membrane of the mitochondria, which is called coenzyme Q. Coenzyme Q is also called ubiquinone. It is basically the cholesterol derivative, and it is the only lipid which is found in this electron transport chain or oxidative phosphorylation. After it we have 3 cytochromes, which includes complex 3, which is cytochrome B and cytochrome C1. At the end we have complex 4, which has cytochrome A3 and cytochrome A. In the middle of complex 3 and complex 4 we have cytochrome C. So these 3 are basically cytochrome. So now let's discuss what is cytochrome and what is its function. Cytochrome is basically a protein in which heme group is present. The main function of it is that it grabs electron and release electron. By grabbing it, it converts Fe3 plus to Fe2 plus, and when it releases, then again Fe2 plus converts itself to Fe3 plus. Now let's start the first process of oxidative phosphorylation, which is oxidation. The process starts with NADH and FADH2. NADH and FADH release its electron in complex 1 and complex 2 respectively, by converting into NAD plus and FAD plus. NADH and FADH2 comes from different processes like glycolysis, which happens in cytoplasm, citric acid cycle or Krebs cycle, which happens in mitochondria, fatty acid oxidation, which happens in mitochondria, and there are different shutters in mitochondria from where the molecule comes through cytoplasm to mitochondria. There are two types of shutter. One is malate aspartate, then there is glycerol 3-phosphate. Malate aspartate is a way for NADH, and glycerol 3-phosphate is a way for FADH2. It comes from cytoplasm. So now let's put up gears and look at through how the process starts and it ends. So firstly, as I told you that NADH and FADH2 releases electron in complex 1 and complex 2 and convert itself into NAD plus and FAD plus. Then the electron goes to coenzyme Q, and after it goes to complex 3. After it goes to cytochrome C and complex 4. Then it moves into the inner membrane. Basically, in complex 3 and cytochrome C, the iron grabs electron and convert itself into Fe2 plus. Then it releases its electron by releasing the electron to further cytochrome and convert itself into Fe3 plus. Then at the end there is complex 4, which uses copper instead of iron and also called cytochrome oxidase. It is called cytochrome oxidase because in it oxygen becomes more negative and it pair ups with hydrogen to become water. Now let's see one condition. If there is less oxygen in the body, the condition is called hypoxia. Then there will be less production of ATP because the process will not go on. So this was the first step of oxidative phosphorylation, which is oxidation. Now let's move on to the phosphorylation and check out how the phosphate add into adenodiphosphate to become adenotriphosphate. There is one more complex which take part in phosphorylation, which is called complex 5. Upper part of it is called FO and the lower part of it is called FI. It is basically a ATP synthase in which it converts adenodiphosphate to adenotriphosphate. But how it happens? That's a big question. So let's see. Previously we discussed that the electron moves in the complexes and comes inside of the mitochondria. When the electron moves, it creates electric current which pumps the hydrogen out of the inner membrane. It creates a gradient of hydrogen, more gradient of hydrogen outside the membrane, so it tries to come in, but the membrane is impermeable to them. But no worries, there is another way for hydrogen to come in. As I told you previously that there is in complex 5 is ATP synthase. So ADP will convert into adenotriphosphate and then the gates will open and the hydrogen will come in. So now the concentration of adenotriphosphate in mitochondria will increase. So now it will try to go out of the mitochondria to cytoplasm. It will go out of mitochondria through a pump called ATP ADP antiport. There are some other things that you should remember. As we discussed, the process starts from NADH and FADH2. The NADH creates 3 ATP which pumps respectively complex 1, 3 and 4. FADH2 creates 2 ATP which will pump complex 3 and complex 4. Another question arises that how the process is controlled in mitochondria. It is not controlled by hormone, it is controlled by energy level itself. When the ATP level increases in the mitochondria, the process slows down, and when the ADP level increases in the mitochondria, the process speeds up to make more ATP. Now there is last but not the least that there are two processes which can break down or blocks the oxidative phosphorylation. Basically, one is uncoupling and the second is inhibition. These are the two processes which can disturb oxidative phosphorylation. Uncoupling is the process in which the new pump is added between complex 4 and complex 5, which is also called ionophore. Because of it, the hydrogen will come inside through ionophore but not from complex 5. So it will lead to do oxidation, but phosphorylation will not happen. So there will be less production of ATP. In inhibition, which is a chemical or a drug, it stops complex functioning, which will decrease the rate of metabolic and which will decrease the production of ATP. So this was today's lecture. Please subscribe us, and if you have any question, please see again the video and comment.
But give me readers. I don't really